This is part 3 of our discussion of the immune system, chapter 21 in our Human Anatomy and Physiology text. In the previous section, we talked about the first part of the adaptive immune system, humoral immunity, the production of antibodies by B lymphocytes, and now we're going to talk about the other arm of the adaptive immune system, cellular immunity, and then we're going to talk about helper T cells that are needed for both of those arms of the adaptive immune system. Cellular immunity is mediated by cytotoxic T cells. Those cells' job is going to be to kill infected cells, cells that have been infected with a virus or a bacteria, and also abnormal cells, cancer cells that don't express their surface proteins correctly, foreign cells brought into the body in tissues that are transplanted. Those are the targets of cytotoxic T cells. Other T cells regulate the immune response itself, and those are called helper T cells, among others, and we're going to talk about those in a bit. So, CD4 T cells, or CD4 cells, recognize major histocompatibility complex type 2 on antigen-presenting cells. These are the cells that are destined to become helper T cells, those ones that we need to allow the adaptive immune responses to proceed. So in order to have B cells complete their activation and produce antibodies, we need helper T cells to recognize an antigen on a major histocompatibility complex on an antigen-presenting cell, on an MHC class 2. CD8 receptor-bearing cells, CD8 cells or T cells, become cytotoxic T cells. So they have a different MHC receptor. Instead of having CD4, they have the CD8 receptor on their surface, which recognizes only MHC class 1, which is found on all the cells of the body. Now, once those become activated, they're going to become cytotoxic T cells, but some, of course, become memory T cells, just like helper T cell activation produces some memory helper T cells. Note, on a side kind of, cytotoxic and regulatory T cells and helper T cells are all the names of activated cells. They are no longer naive. They've found their antigen, they become activated and so forth. And so that's the time when we use those terms. If they have not yet been activated, they're just called CD4 or CD8 cells, or sometimes they're called CD4 positive lymphocytes or CD8 positive or plus lymphocytes. Okay. T cell activation and effector function depend on antigen presentation. When we talked about humoral immunity, we saw that B cells, when they encounter their antigen coursing through the fluid in a lymph node, for example, the lymph fluid, the antigen comes in contact with the B cell that recognizes it finally, and we have activation. Well, in this case, the T cells only recognize antigens that are being displayed on the surface of a cell on its MHC complexes. So there's two types of MHC complexes, as we've said before. Class 1 MHC proteins are found on all the cells in your body, except red blood cells. And class 2 MHCs are displayed on what are called professional antigen presenting cells, right? Because the cells in your body that express class 1 MHC are going to present antigens on there, but the professional antigen presenting cells, the dendritic cells, macrophages, and B cells, produce MHC class 2. That's their kind of calling card for being part of the immune system exclusively. These complexes are synthesized in the endoplasmic reticulum of the cells and then bound to these peptide fragments, and we'll say where those fragments come from, the antigens, and then sent to the surface of the cell. So the cell is now decorated with these MHC complexes with antigen fragments inserted into the surface. So class 1 MHC proteins, where do they get their antigen? Well, they express what are called endogenous antigens. Antigens of proteins that are coming from inside the cell, either in the process of replicating an infecting virus or a bacterium that's living inside that cell. So the infected cell is taking some fragments of those of the proteins being produced by those cells, and displaying it on their surface along with all their proteins that they're making. So the cell has got on its surface this amazing sort of junk pile of peptide fragments on MHC class 1, some of which in an infected cell will be foreign antigen fragments as opposed to self. So this is how CD8 cells get activated. They have the ability to recognize MHC class 1 with their CD8 receptor, and they have a unique T cell receptor, right? Just like B cells have a unique antigen receptor on their surface. Well, T cells have a unique receptor that recognizes a specific individual antigen or antigenic domain, and it's called a T cell receptor, and it will recognize the MHC and at the same time scope out the antigen. And if it recognizes the antigen, the cell becomes activated. Once activated, the cytotoxic T cell will then go around looking for that same antigen fragment on cells in your body on their MHC1, and if it finds it, it realizes it's an infected cell. That's its kind of mission. Class 2 MHC proteins display exogenous antigens. These professional antigen-presenting cells have phagocytized something foreign, bacteria, for example, at a site of inflammation, and digested it with their lysosome, as we saw way back in the innate immune system, and then displayed fragments of those pathogens on their MHC class 2 on their surface. So instead of coming from something that's infecting the cell, the antigen is coming from something that was gobbled up by the antigen presenting cell. And now it's out there on the MHC class 2. And MHC class 2 is what's recognized by CD4, and that is the CD4 activation or T cell receptor activation is going to produce an activated helper T cell. Here's a little cartoon or a table sort of reviewing all these things. Class 1 MHCs are found on all the body cells, all nucleated cells, and red blood cells, of course, have no nucleus, so they're not in that group. Naive CD8 plus cells, as well as already activated cytotoxic T cells, are going around looking for, with their CD8 receptor, the MHC class 1, and then they have this T cell receptor that's looking for a unique specific antigen fragment on that MHC. What kind of antigen fragment? One coming from inside the cell from, say, a virus that's being replicated inside this infected cell. That's what we're going to find on a MHC class 1 in this category. So class 2 MHCs are expressed on antigen-presenting cells, professional phagocytes, and here's a dendritic cell that's gobbled up perhaps it's gobbled up a bacterium, digested it, and on its surface is the fragment of a bacterial antigen on the MHC class 2, and CD4 cells receptor recognizes MHC class 2, so that's when it knows to check the antigen. Its T cell receptor will then check and see if it recognizes a foreign antigen that was again phagocytized by this antigen presenting cell, and if so, it'll become activated and produce a helper T cell. Here's another way of looking at this process we can see in the subsequent steps. A CD4 cell will be looking for any cell that expresses MHC class 2, so it only interacts with professional antigen presenting cells. And so here's the T cell receptor, the unique receptor for the antigen. Here's the CD4 receptor that recognizes the MHC2, and when the connection is made here, here's an antigen presenting cell with its antigen on MHC2. The CD4 cell becomes activated, proliferates, and produces a clone, right? Proliferation over here. And then the clone matures into effector cells, helper T cells that can secrete cytokines that will rev up or allow the final activation of antibody-producing cells of the humoral immune system and cytotoxic T cells of the cellular immune system. CD8 cells are searching around for MHC class 1, which all cells have, recognize it with its CD8 receptor, but then it'll scope out and see if there's a antigen displayed on that MHC1 that is recognized by its unique T cell receptor, and if so, if the connection is made, then that CD8 cell will become activated. If there's a second signal, it'll produce a clone by proliferation, and it'll mature into a killing cell that can go around killing cells that display that same antigen, infected cells that display that same antigen on their MHC1s. But two things to say, both of these processes lead not only to effector cells, but they lead to memory cell production. So just like in the humoral immune system, we must not forget that we're always going to produce some pre-programmed memory lymphocytes that remain in the body after all is said and done, after the whole infection is eradicated and the whole thing settles back down. We're going to have memory cells left in the body that will allow us to jump into action at the re-exposure to the same antigen that we're dealing with here. The other thing is that there has to be a second signal, some way to validate the activation of these cells before they'll go on to proliferate and produce effector cells. That validation process is called co-stimulation. So T cells can only be activated when antigen is presented on an MHC. That's one step, right? If their T cell receptor recognizes an antigen fragment on an MHC, the appropriate type of MHC, they become activated. There has to be a co-stimulation event, a second signal. Right on the antigen presenting cell, there will be another thing that happens. There will be a co-stimulatory event that will take place. The double recognition, this is something that we're sort of backtracking a little bit before we get back to co-stimulation. Even as we said here in this slide, the unique T cell receptor on T lymphocytes that recognizes antigen fragments on MHCs also simultaneously recognize the MHC. So unlike an antibody on a B cell that just recognizes antigen, these T cell receptors recognize both the antigen, the specific antigen, and also the MHC on which it's being displayed. So not only does a CD8 cell, for example, have a CD8 receptor that recognizes MHC class 1, its T cell receptor has to see that MHC class 1 as well, and then the antigen. And if it recognizes the antigen, boom, we have activation. So that's just a little something unique or extra special about the T cell receptor. So the co-stimulation step, we need that second signal to allow that T cell to go on and become fully activated, proliferate, and form effector cells. And so we'll get to what that second signal is, but what if it doesn't happen? What if a T lymphocyte finds its unique antigen, the T cell receptor recognizes the antigen on the MHC, but there's no co-stimulation, there's no second signal? That cell will shut itself off. It'll become anergic. That means anergy means it will no longer ever respond to antigen. It'll essentially say to itself, that must have been a mistake. We weren't supposed to, it wasn't a true infection in the body, it was some kind of an error, and there wasn't a second signal to corroborate that this is an infection, so we would back off, and that cell will become anergic and it will no longer react. So we've saved ourselves from a potential mistake being made where a T cell receptor recognized something that was either a self molecule or something that became looking like a self molecule by a half-din or something. So we've gotten away with something here. We've saved ourselves from a mistake. However, normally what happens is there's a co-stimulation that happens, and so a cytokine may be released by an antigen-presenting cell or a co-stimulatory molecule which will allow proliferation and differentiation to happen. The proliferation and differentiation steps produce a clone of cells which will then peak within about a week of activation. So it takes some time to get this going, just like with humoral immunity. It'll persist for about a week, long enough to get rid of all the antigens-bearing cells, all the pathogens in the body. But we don't want this to go on forever. It's kind of a rough business, a dangerous business. Activated T cells produce inflammatory cytokines, and we don't want them lurking in the body for long periods of time. And so after the final activation and effector cells are produced, they'll persist in the body for a certain period of time, and then they'll finally die. So it may be a month at the most, and all of the activated T cells are going to die by apoptosis, and only the memory cells are going to remain in case of a re-exposure to the antigen. So here's a little cartoon of the activation of a T cell by a antigen-presenting cell. So here's our antigen-presenting cell phagocytizing a foreign bacterium, well, all bacteria are foreign, and it's going to process it or degrade it by a lysosomal action and display fragments on its MHC class II. But it also will display, if it's activated by this phagocytosis, it will display a co-stimulatory molecule. The co-stimulatory molecule is a ligand for another receptor that's found on the T cell. And so the T cell has a CD4 receptor that recognizes MHC class II. It has a unique T cell receptor that recognizes both the MHC2 and the unique antigen. If it didn't recognize this antigen, it would just walk away. But in this case, it has recognized the antigen. Clonal selection has happened. But it also can bind to this co-stimulatory molecule. It's like a receptor and its ligands coming together that will provide this second signal so that the helper T cell is completely activated and can go on to form a clone of identical helper T cells with the same recognition receptor, the same T cell receptor that we've already clonally selected, right? There could have been 1,000 CD4 cells that came by this antigen presenting cell, and they had the wrong T cell receptor and didn't get activated, drifted away. Finally, we found one that had the right T cell receptor, and boom, we made a clone of it, because that's the one that can help us fight today's infection, and then it can differentiate into an effector cell. So cytokines are going to be produced by the helper T cell once it becomes an effector cell. That's one of its main missions, is to produce cytokines, signaling chemicals that can talk to other cells of the immune system. So let's just look at a couple of cytokines. Cytokines are interleukins and interferons. Interleukin 1 is one particular cytokine that we encountered earlier when we talked about the innate immune system. Macrophages, when activated, produce interleukin 1. And interleukin 1 is a cytokine that can cause the production of interleukin 2 by activated T cells. So T cells that produce interleukin 2, that will drive the proliferation of T cells, and they'll form the clone. Other cytokines that can be produced, there's a whole raft of cytokines, as we'll see in a second. Other helper T cells may produce interleukin 4. Gamma interferon may be produced by certain helper T cells, or by various cells of the immune system. TNF alpha is produced by macrophages. It's a very, very important pro-inflammatory cytokine, just like interleukin 1 is. So those are some cytokines that we talked about before. Here's a table of cytokines. I don't want you to worry too much about it, but I do want to mention something about it. It reviews some things. Alpha and beta interferons are the so-called type 1 interferons we talked about, by which an infected cell, a virally infected cell, could talk to its neighbor, warn its neighbor to block it from replicating a virus and slow or stop a viral infection from spreading. Gamma interferon we talked about is a cytokine that is released by macrophages that will rev up the whole immune response. It will rev up all kinds of immune cells. It will activate the differentiation of helper T cells and so forth. Interleukin 1 is a pro-inflammatory interleukin produced by macrophages. IL-2 and IL-4 are interleukins that are produced by helper T cells that will promote the cloning, as we said, of cytotoxic T cells and B lymphocytes. IL-10 is actually an interleukin that's produced that's inhibitory, and it will modulate the immune response. So just like, for example, way back when we talked about things like aldosterone, which can increase blood volume and blood pressure, and then we talked about atrial natriuretic peptide, which can inhibit reabsorption of salt and water and inhibit aldosterone. We have this sort of check and balance system. IL-10 is a immune suppressive interleukin to kind of modulate things so things don't just take off in a crazy direction and with no dampening whatsoever. IL-17 may turn up in a later slide, which is a cytokine that is pro-inflammatory and is thought to be involved in the propagation of autoimmune diseases. So TGF-beta is another inhibitory suppressor cytokine that helps modulate immune responses. along with IL-10. TNF-alpha we talked about as a very pro-inflammatory cytokine produced by macrophages. So just a bunch of a nice sort of collection or review, a nice table of interleukins and cytokines and what they do. Okay, helper T cells. Effector T cells include helper T cells, TH sometimes they're called, this little subscript. They play a central role in all adaptive immune responses. Both humoral and cellular immunity depend on co-activation, co-stimulation by helper T cells. So we can't have any adaptive immune responses without co-stimulation or a second signal produced by helper T cells. They activate B lymphocytes. B lymphocytes become activated when they encounter their antigen, but they won't become fully activated until a helper T cell comes along and gets them finally activated. B lymphocytes incidentally express MHC class II, which is what helper T cells recognize, and so they can come over and meet up with a B lymphocyte who's displaying antigens. The B cell who's found his antigen then displays it on its surface on its MHC class II, and an activated helper T cell can come along, see that MHC class II, see the antigen, and that corroborates, right? If we have clonally selected a B cell and clonally selected a helper T cell that recognizes the same antigen, yes, we have a real infection going on in the body. And so that helper T cell then will see that antigen on the MHC2 on a B cell and finish its activation, and we'll wind up with plasma cells and antibodies being secreted. Sometimes a B cell can become activated without co-stimulation by a B cell. There's occasionally, this is more rare. There are examples that people have discovered in which a B cell, there's certain antigens that can activate a B cell entirely without a co-stimulation by a T cell. Just so you know, so you're fully educated, but the best thing to remember is that B cells do not go on to produce plasma cells and antibodies without helper T cells. And the reason we know how important this is by what happens when there are no helper T cells. And we'll come to that at the end of the last section of this chapter. It's part 3 of this discussion. We'll talk about that. So here's a little cartoon showing a helper T cell that's already been activated, and now it's on the alert. It comes around and bumps into a B cell that's also already been activated, but not completely. It's the car whose engine is running, sitting in the driveway, but it's still in park. And it's got its antigen now that it had already found displayed on its MHC class II, because remember, it's an antigen presenting cell. Now this helper T cell can, with its CD4, recognize that MHC2, and its T cell receptor recognizes the MHC2 as well as this antigen. It's this unique antigen, and then the helper T cell will release cytokines, so IL-4 for example, like we said, IL-2 or IL-4, which will then complete the activation of the B cell, and it will form a clone. It will be a growth factor that helps it form a clone, and it will make plasma cells and antibodies, and we'll defend ourselves against the antigen. Also, sometimes helper T cells will express a co-stimulatory molecule, a ligand, like a cell surface receptor that can bind to a receptor on the B cell and complete its activation that way. So not always a co-stimulation by a cytokine. All right, helper T cells of the CD8 variety becoming activated. Another kind of helper of T lymphocytes, I'm sorry. So CD8 cells also require a second signal, a co-stimulation by a helper T cell in order to form an actual effector cell. So let's take a look at that process. Here's an APC, an antigen-presenting cell, activating a helper T cell. The helper T cell sees the MHC class II, sees the unique antigen. Now it's activated. There would be a co-stimulatory molecule here, and a CD8 positive lymphocyte, right? It recognizes MHC class I, and there we see it. Its CD8 receptor sees this MHC class I here on the APC, and it has a T cell receptor, and it has discovered, in this case, we have clonal selection happening. We found the right CD8 cell that recognizes today's infection, recognizes this antigen, and the helper T cell will then release a cytokine which will drive the proliferation of this cytotoxic T cell, allow it to go forward, proliferate, and form mature effector cells that can go around and kill infected cells in the body. So helper T cells have this role of co-stimulating or providing another signal to drive the final activation of both B cells and cytotoxic T cells, humoral immunity, and cellular immunity. Helper T cells release cytokines that are also involved in other processes. They activate macrophages to make them more potent killers of bacteria. We saw that macrophages need some special tricks sometimes, such as respiratory bursts to kill the bacteria they phagocytose, and T cells will release some cytokines that help rev up that process as well. They'll also release cytokines that will help draw more lymphocytes and macrophages and neutrophils to the site of the infection. So helper T cells have quite a broad role in overall the immune response. Not all helper T cells are the same. They have different names based on the type of cytokine they secrete. And then the type of cytokine they secrete determines what kind of process they help with. So some helper T cells release cytokines that mainly activate cellular immunity, like we saw in this diagram. Some helper T cells mainly activate B cells for humoral immunity. Some helper T cells activate the secretion of IL-17, as we saw. So they're just named for the type of cytokine they produce and the type of response that they get from other cells of the immune system. We're not going to worry too much about that. We will mention something about immunoglobulin E later on, but that's not to worry about that right now. So cytotoxic T cells, we're finishing up our discussion of cellular immunity. How do they actually do their job, their effector function? So they travel around the body. Right? They're going to go find infected cells. That won't work if they're just lurking in a lymphoid tissue like a B cell. They have to go out on the road and scope out cells in your body, and they're looking for MHC complexes, and they're looking for their own unique antigen receptors target antigen on those MHC1s. They will target cells of your body that are infected with viruses or bacteria or parasites, right, that have taken those endogenous antigens from those internal infectious agents and displayed them on the surface on MHC1, and they'll kill them. Cancer cells, a lot of cancer cells display their MHC1 in such a way that the cytotoxic T cell will respond to them and kill them. Foreign cells, if you have a cell that was a tissue that's transplanted into your body, cytotoxic T cells will recognize that they're not appropriately displaying MHC1, the wrong MHC1, and they'll be killed. How do they do it? When a cytotoxic T cell recognizes this antigen and the MHC1, it's going to exocytose two products. It has these products ready and waiting in vesicles. Now it's going to exocytose them, perforins and granzymes. Perforins are proteins that self-assemble into a pore, sort of the way the membrane attack complex of the complement system works. A whole bunch of perforins, when they're released from the vesicle, assemble into a cylindrical pore that stabs itself into the membrane of the body cell that's infected. And then granzymes are small molecules that can diffuse through the perforin pore into the cell and trigger cell death. So we punch a hole, send in some poison, activate cell death, and now that infected cell is terminated. Also, cells express on their surface a sort of like a ripcord for emergency cell death activation. A Fas ligand is called on a Fas receptor on its surface, and cytotoxic T cells can express Fas ligand, which is a compound, a membrane protein that can bind to the Fas receptor on the body cell, and that starts some internal signaling that results in cell death. So it's another way that cytotoxic T cells can kill an infected cell once they discover that the cell is displaying the antigen that they recognize. And here's a little cartoon of it. We can see a cytotoxic T cell surveilling or patrolling the body. comes across a target cell that is infected or a cancer cell and snuggles right up onto that cell, right? It has CD8 receptors that bind to its MHC1 and kind of cling to that cell. And then the cell, if it recognizes its antigen, will exocytose perforins, which form the assembly of these pores, and granzymes are these little dots that then diffuse into the target cell, the cancer cell in this case, and kill it, activate apoptosis. Natural killer cells we encounter when we talked about innate immunity, and natural killer cells are very, very similar to cytotoxic T cells. It's just that they don't undergo clonal selection and activation and those sorts of things, but they kill their target cells the same way as cytotoxic T cells. They recognize abnormal cells, foreign cells, cancer cells that don't express their MHC class I correctly. And so if they bind to those cells, they will also release perforins and granzymes that will kill that target cell. So they're part of the innate immune system, but now it's discovered that natural killer cells have more and more sort of adaptive kinds of functions. It turns out to be more complex than we thought. But anyway, they can kill a similar population of target cells as cytotoxic T cells by the same mechanism. There are, in addition, another population of T cells. We talked about helper T cells, CD4 cells, cytotoxic T cells, CD8 cells. Regulatory T cells will be produced also that are going to dampen down the immune response, compete with some of these activating pro-inflammatory cells to sort of modulate the immune response so it doesn't go entirely off the deep end. They secrete cytokines like IL-10 and TGF-beta, which inhibit a lot of these immune responses or activation processes. Not stop them, but just slow them down, subdue them a little bit. Here's a summary of all the adaptive immune system responses that we've been talking about. So over here, we see an infection. We see some antigens coming in the body. We damage a first line of defense, and some bacteria get into the body. And we have inflammation happening. And we have some macrophages or dendritic cells gobbling up some foreign antigens, some bacteria, processing them. And then and there's probably going to be some dead white blood cells and some damaged bacteria and all kinds of debris hanging around. And the antigens from up here, I guess I should have been pointing up here, come wafting through the lymph fluid or other body fluids where the B cells are all lurking in their follicles, and we clonally select. Once we find the right B cell that happens to have a unique receptor that binds to the antigen of the day, that cell will become activated. And if it receives a second signal from an activated helper T cell, right, co-stimulation happens, then that B cell, that activated B cell, will go on and be put in drive the car, proliferate, mature into plasma cells and produce antibodies, which will inactivate and precipitate and opsonize our antigens, and they'll be gobbled up by macrophages and cleared from the body. Over here we see an antigen presenting cell that's going to gobble up either an infected cell, the whole infected cell, or possibly a bunch of bacteria, and display processed antigens on its surface. It will display processed antigens on its MHC class II, which will be recognized by CD4 cells, which will become activated, co-stimulated, and then proliferate and produce cytokines that can activate B cells. Or the CD8 cells will recognize processed antigens on the MHC1 here and become activated, and the activated helper T cells will produce cytokines that can, or co-stimulatory molecules that can activate this CD8 cell and turn it into a cytotoxic T cell effector, which can go on the road and kill infected cells. Cytokines from helper T cells also go on to activate APCs, macrophages, and so forth, promote inflammation and help the whole system get revved up to another level so that we have everything happening as needed to protect the body from disease. So when we come back, our next session, our final section for the chapter 21, the immune system, will be to talk about a little bit about grafts. Then we'll go on to talk about some abnormalities of adaptive immune responses. Sometimes the adaptive immune system is missing something or is induced to lose something so that it can no longer perform its functions. We have what are called immunodeficiencies. Sometimes, again, they're genetic in nature, sometimes they're acquired, such as when someone gets HIV/AIDS. And then we'll talk about some hypersensitivity, sometimes when the immune system is oversensitive and going off the deep end and producing some side effects that we really don't care for very much. And that'll be the end of our discussion for the most part of Chapter 21. So I'll see you next time.